Hi, I'm Stephen Baccus, and today, together with Rachel Wong, we're going to be talking about the tight link between connectomics and computation in the retina. To review the basic cell types in the retina, signals travel from photoreceptors to bipolar cells to ganglion cells, and along the way are modified by inhibitory cells, horizontal cells, and amacrine cells. So even though there are 5 basic cell types in the retina, there's more than 100 subtypes, 15 or so bipolar cells, more than 60 amacrine cells, more than 30 ganglion cells. A great simplification is that each of these cell types tiles the retina, forming a mosaic of the entire image, which focuses the question on how retinal computation occurs on the interactions between cell types. So we thought that before talking more about the anatomy of the retina, it was important to talk about what the retina does. So a lot of what we might think about what the retina does falls in the category of data compression. So simple filtering centers around receptive fields, also adaptation to various statistics such as luminance, contrast, and patterns. With respect to adaptation, a lot of this comes from mechanisms other than connectivity, such as biochemistry that implements luminance adaptation, synaptic dynamics, and membrane biophysics that implement contrast adaptation. So mostly we're not going to be talking about these types of things that the retina does today. So there's another class of functions that we can think about as computation. Rather than packing all the bits of information up to the higher brain to let the higher brain do the complicated stuff, computation would be extracting important bits of information to drive behavior. And so we can think of these in 2 different classes, one where visual stimuli are detected or discriminated between, or also where the retina can make predictions. So a few examples of discrimination. One is the well-known direction selectivity, where an object moves in one direction, causes firing in retinal direction selective retinal ganglion cells, whereas if it moves in another direction, the cells are silent. Object motion sensitivity, where cells don't respond to global motion as might occur from eye movements, but are sensitive to differential motion as might occur from an object moving in the background of eye movements. When the entire visual field shifts rapidly, as might occur during a fast eye movement, there's a brief burst of inhibition in certain ganglion cells that is thought to be the basis of saccadic suppression, some part of saccadic suppression. Other computations such as orientation selectivity, special responses when when a moving object reverses, cells that reverse polarity. In the predictive category, one computation is called motion anticipation, where a population of cells will shift its activity in the direction of movement, and this is thought to compensate for processing delays when objects move. An interesting phenomenon called the omitted stimulus response, where in response to a periodic stimuli that suddenly ceases, cells will fire a predictive burst of spikes at the time of the expected missing stimulus. Another computation that we've studied is called sensitization, where in response to a strong temporary stimulus, cells will subsequently elevate their sensitivity. And we can explain this as a prediction of motion in response to the fact that, in response to the expectation that when an object moves, it's likely going to be somewhere nearby in the same location in the future. So I go through this partial list of computations to let the retina justify the existence of all of these 100 cell types or so. So these many cell types produce many diverse types of computation. Okay, so how do they do that? So one abstraction that's been used in the retina is the concept of a visual feature. In terms of a circuit that we can see here at left, we can abstract that to a set of synaptic weights, and we can make a spatial map of those synaptic weights here. So this would be what we would think about as a receptive field. So if we include the temporal processing that would occur due to synapses and membrane biophysics, then this spatiotemporal receptive field, in addition to a threshold that might be found at the bipolar cell synaptic terminal, would represent a visual feature, sort of a lower level element of computation. The model that would produce this would be called a linear nonlinear or LN model. So now if you take these visual features and combine them with each of many different cells having many different visual features based on its membrane properties and synaptic input, then these features can be combined to produce computation. So for example, in the case of object motion sensitivity, if there's suppression, inhibition from the background, so bipolar cells would have one type of visual feature, and amacrine cells with long axons would have a different type of visual feature, and combining them together would produce this computation. So this overall organization motivates us, with understanding that different cells produce visual features, it motivates us to pay attention closely to the connectivity and the full connectome, because understanding that connectome and the visual features can tell us how they're combined together to produce computation.
So as Steve has mentioned, understanding the circuitry in terms of the details of connectivity is really important in trying to understand function in the retina. And so I'll just be spending a couple of minutes to talk about what we've learned from retinal EM connectomes so far, what we gain in terms of knowledge, and what else do we need. But first, let me remind everyone that, in fact, transmission EM has over many decades now been very instrumental in reviewing the synapse architecture as well as connectivity of cells across the retinas of many vertebrate species. But really, semi-automated serial EM methods more recently has enabled more complete reconstructions of very much larger tissue volumes. And this has greatly assisted cell identification and classification, and which of course together has led to the discovery of new cell types, particularly in the mouse retina, and also novel circuitry. Now, let me just illustrate a little bit about why dense reconstructions can aid the classification of retinal cell types, just by considering one class of bipolar interneurons, the type 5s. Generally, both cell morphology in terms of cell size and branching patterns, as well as the stratification of the axons and dendrites of retinal neurons within the depth of the inner synaptic layer, are very good hallmarks or indicators of a particular cell type. But sometimes it's really difficult just based on stratification and even morphology to define and separate whether these are in fact the same or a common cell type. If you do a dense reconstruction of all the axons, in this case, that are small field and stratified within this region, and apply a fundamental organizational principle of the retina, and that is individual cell types tend to form cell mosaics and their arbors tile, then in fact, you can separate out this almost messy population into 3 distinct subtypes, in this case, subtypes of type 5 bipolar cells. Now, serial block phase EM, as well as other methods of volume reconstruction, have also been instrumental in identifying unusual cell types in the retina. For example, there's a cell that stratifies very close to this type 1 bipolar cell, but it doesn't even look like a bipolar cell, it's monopolar. However, it's glutamatergic, and it has all the hallmarks of a bipolar cells, especially containing ribbon synapses. So this is now called type 1b, it's kind of half a bipolar cell or a gloomy cell. And not surprisingly, of course, we've identified many more features of circuitry that underlie specific functions in the retina, especially for direction selective ganglion cells. As Steve has mentioned earlier, we know a lot about these cells, and we also know that direction selective ganglion cells achieve this sort of feature by virtue of their asymmetric connections with these inhibitory starburst amacrine cells getting greater inhibition on one side versus the other. However, more recent experiments that combine both calcium imaging of the ganglion cell layer and serial block phase reconstructions of this area has helped us discover that different parts of an individual starburst amacrine cell is connected to different ganglion cells. And each of these ganglion cells actually are direction selective, but they encode for different directions of movement. And so now you get a more complete picture of how the circuitry can help encode different directions, in fact, meeting these 4 cardinal directions. It's not surprising that connectomics have also revealed unconventional contact and synapse arrangements. For example, cone bipolar cells talking to rods and rod bipolar cells talking to cones. And my favorite example is actually this one, because it breaks almost the synaptic connectivity rules of the retina, because normally axons and dendrites of synaptic partners would co-stratify. But in this particular cell, the M1 melanopsin ganglion cell, its arbor is actually in the off sublaminar, but it's receiving connections from this on bipolar cell, which stratifies in the on layer, and it receives connections from the axon shaft. So this is rather unusual connectivity between 2 partners that actually do not meet really in terms of the density of their space. Well, with this and even more examples that we don't have time to go to, we've learned a lot about circuitry, but we need even more, I think, complete maps. So what else do we need? There are already volumes that encompass potential possibilities of contacts from photoreceptors all the way to the ganglion cells. This is through contacts defined by membrane appositions. Volumes that, in fact, have identified synapses, so later volumes, in fact, only actually encompass or cover synapses or connections in the inner retina. So we are still missing a lot of vertical connections. Like other parts of the brain, where there are also lots of horizontal connections, there are also these long-reach endocrine cells that Steve talked about earlier, and they're very, very large. And the largest volume we have so far only can capture a small part of these cells. And therefore, we need even larger volumes to capture all these horizontal connections and get a more complete map of the circuits. The other thing we need to really, really consider is that the retina, in spite of the fact that we always say it's simple, it has repeatable units all across the retina, these units actually can vary quite a bit across different regions of the retina. So we have to consider cell distribution, connections even, and visual responses, which may change because circuitry is different. So 2 examples here. A particular ganglion cell that is very transient in its response to darkness, step of darkness here, become more sustained in its response depending on where it sits. Also, there are cells that are direction selective if they're sitting here, but the same cell that's molecularly defined actually is not direction selective when it's sitting in the more peripheral retina. We know a lot about distributions of cells not being uniform. Classic one, of course, is in the mouse retina, where cone photoreceptors that are short wavelength or long, medium and long wavelength are actually distributed across the retina in opposing gradients. And I'll really include the connectivity of cells can also change. And this is partly because, for example, ganglion cells of one type could be small in one region and much larger in another. And so can the axonal territories of their presynaptic cells. So there's variation in cell size and possibly connectivity. But perhaps the most dramatic example is this one, the midget circuitry, where in the region near the fovea, within the fovea, individual cones connect to one of 2 on and off bipolar cells that connect to a single ganglion cell, enabling these ganglion cells actually to have single cone resolution. This sort of very tight circuitry with little divergence and convergence is very different from what's seen from the same components of cells in the peripheral retina. This example also highlights species differences, where in the where we see midget circuits that are in the human retina and non-human primates, but not, for example, in mice. I think it's also important for us to really consider connectomes and obtain more of the connectomes of human retina. And I think EM approaches are probably the only way to do this because we don't have the luxury of molecular approaches or genetics to help us identify cell types and their connections. And I think getting connectomes for human retina is really important for some obvious reasons we'll describe next. And connectomes also the connectomic approach can also be used to help us study the development of human circuits like the fovea circuits in this example shown here. So finally, we think it's very important to also get what is Robert Marks calls the petoconnectome. And this is because circuitry in the retina is actually remodeled or rewired quite differently when you lose, for example, photoreceptors. Studying and assessing what this rewiring actually looks like, I think it's very important to help strategize how to repair circuits, whether it's through cell transplant therapy or through prosthetics. And importantly, we can also use, I think, zero-EM methods to study how circuits might reassemble, especially when you're able to transplant new cells or create new cells from endogenous sources such as this one, converting glial cells into neurons. And what better way to actually determine how well neurons integrate into the disease environment, perhaps reassemble the circuitry by using connectomics and also correlating that with physiology. So what I think I've done here is hopefully given you a sense that if we were to collect more volumes, we really should need to capture the rest of the connectome, vertical, horizontal connections, and consider topographic species differences, need to do more for the human and non-human primate retina in order to strategize vision restoration. And to do that, we need larger volumes. We need to get volumes at different locations, of course, different species. And what I haven't talked about, and Steve will now carry on to bring out this topic is to try and match these structural connectomes that we can get from serial EM data with circuit function. And I'll leave him to tell you more about that.
So how are we going to connect the connectomic structure to function? So a potential tool is a type of model that we've used successfully to capture visual computation in the retina. So this is called a convolutional neural network model, a machine learning approach that is widely used. And that's because each of its basic elements are the same visual features that we talked about. At each location, this little motif visual feature is presented across the visual field, and that forms the spatial convolution, just like a mosaic of retinal neurons. Now, even though in this typical case, this is uniform, as Rachel mentioned, there are non-uniformities in the retina, and those can be built in as well. So the types of model that we have used are very simple, small types of these models, 3 layers, like 3 layers in the retina. And so each of these convolutional layers within them, there are different cell types with different filters, just like the different cell types in the retina. So 8 cell types in each of the first 2 layers here. With this model, we've been able to capture the response to really any arbitrary visual computation or visual stimulus. So including natural scenes or natural movies, the data in the model are correlated to nearly up to the retinal variability, and much more so than other leading types of models. Not only do these models accurately predict the response, but if you look at their internal units, they have model units that are highly correlated with our own recordings of interneurons that we've made separately. And they also reproduce a broad range of computations that I've talked about when only fit to natural scenes. So the fact that the model units are correlated with interneurons tells us that these models can be not only informative about computation, but also connect to structure as well. So if we know that the retina has retinal structure and cell type biophysics that leads to visual responses, an important goal would be to create a model that both incorporates the connectome, the cell type specific biophysics, and produces the visual responses. I say this is an intermediate goal because the real goal would be to use this tool to answer questions about how mechanisms produce visual computation and to test theories of retinal function. So I'm going to end here with 3 potential new approaches that we can use connectomic data to understand retinal function. The first would be to take one of these models and fit it to natural scene responses, which we know will capture a broad range of computation, and then just inspect the model to see whether or not the connections within the model match the measured connectome. So there are many potential models that can capture the data. So with this approach, we can ask which type of models are actually informative about anatomical structure. Do we need models that have the recurrent circuitry within amacrine cells incorporated into it? Do we need to add multiple layers that would incorporate bipolar cell to amacrine cell to ganglion cell transmission? So this can focus us on different types of models that can capture both function and structure. An additional approach would be to, from the very beginning, build in the connectomic data. So train models to match the measured connectomic data and the measured cell-to-cell type connections, and then train it to fit visual responses, either something broad like natural scenes or specific computations, and then use such a model to ask how those specific cells create the computations. And we already have the tools to do this, to analyze the model, and at each point in time say which cells are contributing to each individual computation or response. And then a final approach would be to take known structural data and known biophysical data, and which in some cases can come from preparations where both are done, and then just fix it into the model to begin with. And then ask, does such a model actually predict visual responses? And so to be clear, what this means is, can we take known structural and functional lower level data and use it to explain emergent neural circuit function? Now, just to point this out, if this can occur, this would be a really surprising and important development, because in any kind of system, it's very hard to take the lower level function and predict higher level emergent behavior. So, but I think we're getting close to be able to at least test whether we can do that. And so in summary, these new approaches with connectomics really come from the fact that there is a very tight link between structure and function in the retina.